Okay, so I thought we'd start by changing things a little bit. Let's do this question. Let's work on this question, see how far you got, and I'll go through the solution. Then I think I'm going to change the order a little bit, and we'll start talking about virus filtration, because I think that fits in better with the flow of things. And then we'll talk about membrane adsorbers this afternoon. And in the meantime, you will have two lectures, one on, I think, electroprocessors, electroactive membranes, and then one on biofouling. Okay, so who wants to tell me? 1 second. Who wants to tell me how to solve this problem? Anyone want to volunteer at least a solution method? Because the main thing is to see if you know the method of solving this. So who wants to suggest what we would like to do? Actually, better still, who would like to draw a diagram of what's happening? Would somebody like to come and draw a diagram of the setup, what's going to happen, right? So you have this unit of red blood cells. So who would like to draw a diagram? Volunteers? Anybody? Anybody? Somebody must have thought about it. Who would like to do? Who's going to volunteer? Somebody. You want to volunteer? Okay. Yes, come. You can draw the diagram. That's easy, right? So go ahead. I guess we can draw it on the question. That's fine. So then everybody knows what we're trying to do. So just the diagram of what we're doing here. Okay. Okay, so that's our microfiltration part. But can you draw the whole experimental, the setup? So your bag of red blood cells and how we're going to pump around and so forth. See, you are in red blood cells, right, to be concentrated. So can you draw the bag of red blood cells? Go ahead, draw a bag. A bag, which is a tank, the red blood cells. Just a bag, a tank, just a tank. That's fine. Just draw a tank. Yeah, good. Now from there you're going to pump, right, through the module that you've shown here, right? That's okay. So come, just put the line down like this. So I did it already. Never mind. Okay. And then what happens? What happens now? This is then from the module, the retentate. Where does that go?
That goes to
yes. So we can do that. Just put that going back there. Good. Okay. And we're going to concentrate, and there's permeate that comes out here into a container, right? So that comes into a container. Good. All right. Very good. Yeah. So basically, I'll redraw that. Just a second. Okay. So redrawing that. Okay. So this is what we have for the system, right? Which he showed us basically. We have our blood bag. We're going to pump. Actually, the way you run these typically is, blood bags. Have you seen them? They have a port at one end. So this is actually held up here. So it's upside down. So the port is coming out of here, okay? But you come out of here, you go through the module, which could be hollow fibers, it could be flat sheet, we don't say. The retentate goes back into the bag, right? And you're collecting permeate. So that's the first part of the problem, right? And so you're going to start concentrating this blood. So who can tell me, we start off with a volume of 1000 ml, right? At a hematocrit of 15%. And we want to concentrate it to a hematocrit of 0.4, okay, 40%. So how much liquid we need to remove? Who can tell me? Who had a look at this and worked it out? How much liquid we need to remove from this? Anybody know? In other words, what's the permeate volume? That's a fairly easy question, right? I hope. Can anybody tell me how to, it's a mass balance. Anybody tell me how to calculate it? Sorry? 150 ml you want to remove. Okay. So you're telling me, so how would you do it? So tell me what to write. I want to write an equation.
50% of the initial concentration.
Okay, so hang on a second. So initial concentration is 1,000, right? And you're saying to take 15% of that. Okay, so multiplied by 0.15. Okay, that's equal to 150 ml of what?
Initial.
Red blood cells, right? These are red blood cells, right? Okay, RBCs. Okay, I agree. Then what?
Next, we have to get it to 400 ml.
You have to get it to, why do you have to get it to 400 ml? You have to get it to a constant, you've got to get it to a concentration of 40%. Okay, all right. So you're going to say now, you want 40% of 1,000 ml. So 0.4, right, multiplied by 1,000. And so that gives me 400. Okay. And so what does that do? So this part I agree with. I'm not sure about this one. Okay. 375 is what? 375 is a number, but what is 375? The amount that you remove or that is still in the bag? Correct. 375. So can you tell me how you got that? Correct. Correct. So hang on, we had that here already. 1,000 multiplied by 0.15, yeah, equals?
X volumes into 0.1.
Correct. And so that equals actually what you got, right? 375, right? So if it's 375, we need to remove 625 ml, right? That's what you need to remove. Okay? Correct? Everyone agrees? Good. Now, what is the question asking us? So that's okay, but don't forget what the question is asking. The question is saying, what size hollow fiber module is required? In other words, what sort of membrane surface area? So, now we know how much liquid, sorry, my ml is not good, we have to remove. So, we know the following, that we have to remove 625 ml, and we have to do it in how long? It told us, right? Did it tell us? I thought it told us. You may assume that centimeters. Didn't it give us a time here? Now I can't see. 10 minutes I thought, right? But why can't I see? Second line. Oh yes, okay, good, thank you. Yep, 10 minutes. Okay, so you need 625 ml in 10 minutes. Okay, so we need to figure out what membrane surface area we need. Okay, that's the next point. So how are you going to work that out? So what do you need to know now? So you know the membrane, you know the amount you got to remove, you know how long you have, and you know you now need to come up with a module with a given area. But what do I need to know before I can go any further? Sorry? Flux, very good. And so do we have an equation for flux? So I'm going to clear this, right, because I think everyone knows this. So do we have an equation for flux? Well, I gave it to you yesterday. Okay, I didn't go into all the theory, but you wrote it down yesterday, right? So the flux equation was the following. J equals 0.078, right? Is that right? A, I think we called it A to the 4th divided by L. All this to the 1/3 gamma dot. And then there's the wall concentration, right? Is that right? Divided by the bulk concentration. Is that correct? And there's a log term here. Is that correct? Okay, good. Now, do we know everything here? So, what, do we know this one? Yes, we do. We're told that somewhere, right? 2.6. Remember the units, though. It's micron, okay? 2.6 multiplied 10 to the minus 6. L, the length of the module, do we know? Yes, 10 centimeters, 10 by 10 to the minus 2, right? Good. Then the concentration at the wall, we are told, is actually, since it's a ratio here, you can use a ratio. So this is 0.98, right? So we know all this. Now, to determine the flux, we need a value for CB. But the bulk concentration, we could use 0.15, right? And if you use 0.15, you will get a flux. But ultimately, we're going to end up at 0.4, right? That's the final concentration. Another flux. Is the flux higher or lower at 0.4 compared to 0.15? Sorry? Lower. Yes. So you can say it from the equation because this log term will be smaller, right? But also think of it qualitatively. If your cell concentration goes up, your flux goes down, okay? That's something you should know. If the concentration of what you're trying to process is increasing, the flux goes down, okay? That makes sense, right? Because you get more cake formation and so forth. So that's one thing. If the viscosity, for example, goes up, the flux goes down. These are just qualitative things you should know without even having to do any calculations. Okay, so who could tell me how you would want to solve this? Do you want to use 0.15? Do you want to use 0.4 to get the flux? Or do you want to use something else? Sorry? What do you say? It? Say it again, 0.15 into, I didn't understand, 1000 equals x multiplied by 0.4. When we didn't have this though. Oh, okay, 375, that's fine. But no, the point is you need a value for CB, the bulk concentration, okay? Okay, that's the volume that you have to remove. Yeah, that we know. Okay, I agree. But we need a value for CB. So I'm saying, are we going to use the final value, which is 0.4? That's your final concentration, and we've agreed the flux is lowest then. Or are we going to use 0.15, which is the initial value, and the flux is highest? Or do you want to use something else? So I'm just looking for ways to solve this. Suggestions? Average of what? Okay, so you're suggesting we do 0.15, right? Plus 0.4, and that's 5.5, right? And then divide that by 2, right? Okay, we could do that. That's one possibility. What would be wrong with using 0.15? Imagine you're working in industry. What would be wrong with using 0.15? Or what's right about it? What are the disadvantages of using 0.15? So 0.15 gives you the highest flux, right? Time will decrease. So you know if you use 0.15, probably you're underestimating the time, correct? What's wrong with using 0.4? You're opposite. You're overestimating the time, but on the other hand, you could argue you've got a factor of safety and so forth, right? You could do what she says and use the average flux, and that might be a good approximation, right? Just one thing to remember, that there is a mathematically rigorous way to do this, and that would be to do something like this, to take the flux, right, and integrate it with respect to the concentration and divide it by this, okay? So all of you who are engineers should have seen that somewhere, because who knows Hagen-Poiseuille equation? Nobody? Somebody must know Hagen-Poiseuille. We must have learned Hagen-Poiseuille equation, right? So do you remember how you derived it? You derive it by doing these, looking at these laminar, these layers, if you like, flowing through, and you integrate, right, through the cross section of the tube. Same sort of thing we're doing here. So you can find a mathematical average, okay? And if you like, we can just post the solution. I don't want to go through the whole thing, but it can be done. And if you did that, you will come up with a number which I worked out here. But I agree with both this answer. I think that's fine. I also think that using 0.4 would be fine, because that then gives you an overestimate of the time, but having a factor of safety is okay, right? So you could do it any of these ways. And also remembering that if you do the actual mathematical calculation, the reality is you might have a few lysed, broken red blood cells, you might have a few other things, which will mean that your flux may be lower than you predict, okay? So all of those are fine, those answers. Okay, did anyone get an answer? Did you get an answer for this using this value? Okay, all right. So I'll let you keep working on that, and then you can tell us what you will get for the membrane surface area. Is that okay? Okay, so while she's working on that, let's the rest of us look at part 2. So part 2 says it is necessary to remove the plasma proteins from the concentrated red blood cells. This can be done by diafiltering the blood using physiological saline. So we have to come up with a new diagram now, right? So let's do a new diagram. Who wants to try a new diagram? Who would like to do the new diagram? So remember, we have this so far. We have, okay, we have this so far. So we have a blood bag here, right? We have something coming out here. We're going through our module. We have retentate coming back in here. And we collected our permeate, and we collected whatever it was, 625 ml. So we have 625 ml here already. Now we're going to stop and want to do diafiltration. Who wants to, we're not going to change this module, okay? It's the same module. So who wants to draw the extra loop for the diafiltration? Volunteer from this side maybe, since we've had everyone from that side. Who would like to draw the loop for the diafiltration? Anybody? I need someone to draw this. Volunteer, somebody. Who wants to? You want to? Okay, go ahead. I will give it a pen, and you can draw the part. So we have to add something to this, right? This is the setup we have. So hang on a second, hang on. I have a question. So what are you trying to do here? So
You got the permeate, okay?
yeah.
So from that concentrated red blood cells, I'm trying to.
The concentrated red blood cells are here.
Here.
Yes. Remember? Because you're removing the permeate. Yeah. That's fine, yeah. So that's going to be your saline that you're pumping in, right? And so probably you need to put a pump here. There'll be a pump there, that's okay. We didn't put it before, and there was also actually a pump here because you were pumping through this. So there are 2 pumps now, right? That's it, no, that's it, nothing more. That's it, yep. So can everyone see that? Basically, we have our saline here, right? And we have a pump here. So this was always pumping around this way, and we were collecting permeate. And now we have a pump here, and we're adding saline. The only catch is we want to add saline at the same rate at which we are withdrawing this one. So one way to do it is that if it's a blood bag, you'll have to pump. But if you had a rigid vessel, okay, you could just attach this here, and because you create a partial vacuum as you're pumping out from here, this will automatically suck stuff in at the same rate. But a blood bag won't work because it'll collapse, okay? All right, so we got that. Do you have an answer yet for our membrane surface area? 22.5. Let me see. 22.5 what, cm squared? Meter squared, no, that's too large. Okay, so, but anyway, thank you for trying. Let me tell you what it comes up with. And then you guys can look at it at home. I suspect it's a unit error somewhere. Okay, that's what I suspect. Okay, so here is what we have to do. So you put in your numbers. So for me, I actually did the whole rigorous method of trying to come up with a flux. Your value would be slightly different, okay? And I came up with the flux of J. Now, we never talked about this before, but typically the unit of flux, so this is Q, if you like, it's a volumetric flow rate divided by area time. So typically in these sort of processes, you would talk about flux, if you're talking about the biotech industry, in things like liters per meter squared per hour, so LMH. That is quite common in the biotech industry, where the volume is in liters, the area is in meters squared, and the time is in hours. Okay, so they are SI units, but they're not standard SI units, but that's typically what people use. Okay, in the blood processing area, they typically don't talk about liters, because I don't know how, if you're aware, but when you donate blood, you donate about, depending on the country, 500 ml, 600 ml, 300 ml, something like. In India, is there a standard? 500, okay, 500. In the US, I think it was 500, they went to 600, but I'm not sure. It depends on your weight as well, okay, the patient. So typically, talking in liters is not making sense, right? Because 600 is 0.6 liters, it's getting, or 0.5 liters. So typically in these areas, we work in slightly different units, and the units are typically something like centimeters per minute. Okay, now how does it become centimeters per minute? Well, look at this. This is actually a volume and this is an area. So actually this is just a length divided by time. So that's how that comes, okay? From a understanding perspective, it's better to think of it in terms of volume per area time, because a flux is always your unit divided whatever it is, mass, volume, whatever, per area per time, okay? But in this case, they cancel out and you can just call it a length per time. So it's a velocity. So anyway, I get 1, and we'll put this solution up, 1.5712 multiplied by 10 to the minus 5 meters per second, which in centimeters per second, then you can centimeters per minute, sorry, is 0.0943 centimeters per minute, okay? So once you know that, then you know J, right? You know the volume you have to remove, you know that V, you know T, you can get area out of that, okay? And the area I get actually is 663 square centimeters, okay? So it's a little different, but I suspect your error was a unit conversion error somewhere. Okay, now if we go that, is the rest of the question very simple or not? What would you do next? I think it's very simple, right? So now all it's asking you is you've got to do a 3 times diafiltration. So you need to know the flux, right? But the flux, the concentration of red cells is not changing. So you could use your C for the blood now as equaling 0.4 if you wanted, right? Because that's what it is. And you know your membrane surface area. And how much volume do you have to treat this time? That's the only question, right? How much saline are you going to add? Anybody? Sorry? You mean 375, right? 375. You're going to add 375 ml of saline, or how much? Look at this. Yes, correct. So you will add actually, as she said, 3 times 375, and I got a time of 26 minutes, okay? So the total processing time is then 36 minutes, because 26 plus 10, okay? So I hope that question was helpful. It brings together a lot of concepts that we talked about, right? So you should have an idea now of how you would design these membrane systems. Yes, so maybe at the end of the lecture, can they put it up? I have it here. Or we can do it now. Okay. So I will give this to you right now, and you can actually take a photo of it, and then we can put that up later, okay? So that was the first part. That's part A. That goes up then in part B. Okay. All right, let's go on then. So that I hope is helpful. Let's go on then with the start of today's lecture. And if you have questions, just ask me during the remaining days, because there are many other questions like this that we could do. But this actually gets a lot of the principles in terms of designing these membrane modules, okay? Because that would probably be the main thing that one would do. Okay, let's talk about, so I wanted to make a small change today, and let me, okay, now I need to project, just 1 second. Yes, good. So I wanted to make a small change. I was going to initially talk about membrane adsorbers right now and virus filtration this afternoon. I'm going to switch the order, because actually virus filtration is also a pressure-driven process, so it fits well with what we've talked about so far, and then membrane adsorbers is something completely different, okay? So let's do it that way. So virus filtration. So let's continue. All right, so just to summarize where we are, well, we've talked about these processes. So we're coming actually to the end of these pressure-driven processes. So we talked about bioreactor harvesting, right? And we said that that could be tangential flow, and it could also be depth filtration, right? And there are some other things I'm going to talk about when we get to emerging therapeutics about developing methods. But in terms of what's established there, we did those two. We talked about sterile filtration yesterday. We talked about ultrafiltration, right, yesterday, right? And so now if you look at it, the only remaining one is this one, virus filtration. So if you remember, now I need to turn this off, right? If you remember, I mentioned yesterday that, well, actually, what did I mention yesterday about virus filtration? Let's see who remembers. So what's the difference between microfiltration, ultrafiltration, and virus filtration? Anybody? Well, let's think of it different. In microfiltration, what are we trying to do generally? Correct. Remove micron-sized things, right? Ultrafiltration, what are we trying to do? Proteins, what about proteins? What do you want to do with them? What do you want to do with proteins? I agree it's with proteins. What do you want to do? Yes. Can you be more specific? Concentration, yes, or removing the buffer, diafiltration. Yes. We will talk a little bit about protein separation that you mentioned tomorrow, okay? But that's a developing method. But today, it's just ultrafiltration, use protein concentration, buffer exchange. Okay, so that's those two. What's virus filtration trying to do? Remove viruses, contaminating virus particles. So I had mentioned that basically virus filtration membranes consist of microfiltration support with an ultrafiltration layer on top, okay? The other thing is they run in reverse asymmetric mode, okay? So microfiltration, the pore size is 0.05 to 10 micron. We want to, as she mentioned, retain cells or get rid of cells, cell debris and other stuff. Then we look generally in dead-end mode for sterile filtration, disposable units. Now we're going to talk about virus filtration, which in a way is between microfiltration and ultrafiltration. Virus filtration, actually, these membranes are actually large pore ultrafiltration membranes, okay? Pore size is between 20 and 70 nanometers. So one thing we didn't talk about, but if most of you are familiar with membrane technology, for microfiltration, we talk about a nominal molecular weight of, nominal pore size of the membrane, right? 0.2, 0.45, whatever. For ultrafiltration, we don't do that. We talk about nominal molecular weight cutoff of the membrane. For virus filtration, we typically don't do any of this. We say whether it's a retrovirus rejecting filter or a parvovirus rejecting filter, okay? But to put it in perspective, the pore size is between about 20 and 70 nanometers, okay? They are disposable units because they cannot be cleaned and revalidated, and ultrafiltration, protein concentration, buffer exchange. So ultrafiltration, the pore size is typically smaller, between 1 and 20 nanometers, okay? All right. So here is our processing train. So we've basically done almost everything. Why is it doing this for me? Okay. We've done almost everything here. So we've talked about sterile filtration, right? We have talked about depth filters. We've talked about bioreactor harvesting, which is over there somewhere. We have talked about ultrafiltration here, UFDF. So we're going to talk about virus filtration. And you must note the virus filtration comes towards the end of the processing scheme, okay? It's always towards the end. And that's important because virus filters are trying to do something that's very tricky. Basically, they're trying to separate viruses from proteins that are very similar in size. So if you have any kind of other species, foulants, host cell proteins, anything else there, it's going to mess up this whole process because you've got a very tight separation to do here. Anything else will plug the pores and you'll end up with a mess, okay? So that's why virus filtration is conducted on very, very pure feed streams. The contaminants that lead to fouling of the membrane are product-related contaminants. That's very important in the biotech industry. In other words, we talked about aggregates, but not huge aggregates, maybe 2 or 3, maybe trimers of what you want, okay? Or these so-called associated species. So they're not insoluble, they're soluble, and yet they are kind of associating, sort of, okay? So it's not really an aggregate as such. Or product variants, okay? Something that are denatured and so forth. Those are the contaminants that can foul this membrane, okay? So they are product related. The virus particles themselves will not foul the membrane for 2 reasons. One is, actually there aren't any virus particles, right? There shouldn't be. This is done as a safety measure to prove that if there were viruses, you've removed them. So in spiking tests, of course, you spike with virus and you test, but in reality, there shouldn't be virus that will foul this membrane. The other reason is, if you were to work it out, even if you have things like, say, 10 to the 6, that's typical. say virus particles per ml. That's what you could have, say, if it were there. You look at the protein concentration. So your protein may be things like 10 grams per milliliter or something like this. It's can be quite high. 10 grams per liter. 10 grams per liter. It can be quite high, but you will convert that, if you knew the molecular weight of your protein, into a number. You will find that it's huge compared to this. Okay, it's certainly less than 1 molar, but it's huge. Okay, remember 1 mole is 10 to the 23, so it's huge. So there's no way that this could lead to fouling. The numbers are too small. Okay. Okay. So let's continue. So regulatory authorities, so this is one of the unique parts about biotech applications of membranes as opposed to anything else, even water treatment, because this is very strict here. Regulatory authorities require validation of what's called adequate virus clearance, okay? So adequate virus clearance is actually a very vague term because it's quite complicated. This is quite complicated, okay? So most engineering applications require scale up, but as I mentioned, I think on Monday, here, accurate scale down is required to demonstrate virus clearance at process scale. So you have a manufacturing facility, you've got that scheme that I showed earlier, and now your job is to prove that with these unit operations that are there, you get enough virus clearance, okay? That is what you have to prove. If not, you've got to put an extra step there somehow. And you don't want to do that because what does that mean? More capital expenditure, more loss of product, because no step is 100% efficient, and so the whole thing gets worse. Okay, so you have to validate this. So you cannot possibly take this whole manufacturing scheme and start spiking it with virus, okay, and then saying, now I show clearance, right? Why can't you do that? Well, firstly, you're going to infect the whole thing, and you'll never be able to validate that you cleaned it. But secondly, but I don't think there's anyone here with a microbiology background, right? Is there? No. If you had, you would know that to grow virus is quite complicated, and to grow it to that number, okay, if you want 10 to the 6 per ml, and you're talking about 10,000 liter bioreactor, that's a huge number. You couldn't do it, okay? It's just not feasible. So you have to scale down, and this is a case where scale down is very important because a small scale unit should give the same results as the large scale, okay? The level of clearance, so clearance can be by inactivation or by removal. It doesn't matter, but it has to be one or the other. You can inactivate them or remove them. Is very high, requiring multiple unit operations. I told you yesterday, that there are two sources of virus infection. So one is the fact that you are using cells, and these cells were modified using something called retroviruses. And if you do the calculation from the inoculum all the way up to the bioreactor, you may need to remove something like 10 to the 15 retroviruses per ml, say. That's actually a very huge number, okay? That's just huge. So if you've got such a huge number, you cannot use one unit operation because you can't grow viruses at such a high titer. So you're going to have to use multiple unit operations and then add them. And then, as I mentioned, they have to be so-called orthogonal steps, and that is because you can't say, okay, I got so much from my virus filter, and I'm going to put 3 of them in series, because the argument would be, okay, so you used a virus filter, you got, say, 10 to the 6 removal, okay, a millionfold removal. That means some went through. So if you use another virus filter in series, it should be less effective, right? Because these ones somehow were able to get through. And so how do you know the next time it's going to have the same efficiency, okay? That's the argument. So you have to use a different method. So size exclusion is one, ion exchange is another one, and so forth, okay? And virus filtration aims to virus particles while in the product of interest pass through the membrane, okay? So it provides robust size, so virus filtration in particular provides robust size-based viral clearance that complements other clearance steps. And actually, we don't talk about them here because they're not really membrane steps, but there's low pH hold I talked about. You actually can also have membrane adsorbers, which we will talk about, okay? And people are looking at other things as well, okay? There are other methods people are looking at as well. Presence of small number of abnormally large pores will permit excessive virus leakage. Manufacturers must eliminate all macro defects. So here's another thing to think about. When you talk about protein concentration, if you can get, say, 95 to 99% recovery of your protein, that's pretty good. That's fine. But virus, so if you think of it, 99%, so if you think of a 1 log as being 90%, right? So 2 logs is 99%, and I was talking like 10 to the 6 or 6 logs. So you see immediately that the level of leakage you can have is much, much less. You cannot let even one virus particle go through. So that's why you have to make sure there are no macro defects. Okay, that becomes very important. Okay. It's accomplished through composite membranes that provide required combination of virus detention and mechanical stability. Multiple layer membranes are often used. Virus filtration membranes are made from hydrophilic polyether sulfone, PVDF, or regenerated cellulose. Those are the most common materials, and they're the same as for ultrafiltration and microfiltration membranes. Now, this is important. So previously, there were so-called parvovirus and retrovirus filters in the purification train. So retroviruses are the ones that are present in very high concentration, and they are the ones that exist because the cells were multiplying and produced viruses, right? And they are typically around 100 nanometers in size. Okay, therefore, these were the ones that you use the 70 nanometer pore size membranes. Okay, I told you they are between 20 and 70 nanometers. Parvovirus are small viruses. They are typically 18 to 22 nanometers in size, and they are so-called adventitious virus infection, and we're looking at removal of about 10 to the 6, about 6 log, okay? And this comes from using animal-derived growth media and so forth, okay? So in the past, you used to put these multiple virus filters, some for parvo, some for retro. So what you could say fairly clearly is that if I had a filter that removed parvovirus, the smaller virus, surely it must remove retrovirus as well, right? Because it's bigger. And that's in fact what happens today. So you don't actually have this today. In fact, you only use parvovirus filters that reject smaller parvovirus, and they are automatically validated for removing retroviruses, okay? And so virus filters are large pore ultrafiltration membranes that basically run in dead end mode with the support structure facing the feed in reverse asymmetric mode, like the biooptimal filter I talked about, okay? And the membranes are made to much tighter specifications than ultrafiltration membranes. For some reason, the bottom part of that has got cut off, but anyway, that's okay. So virus filters, okay, so virus filters originally were designed for use in tangential flow mode with the feed flowing adjacent to the skin surface layer of the asymmetric membrane, exactly the same actually as ultrafiltration. That's how they were originally run. So it was just ultrafiltration. But I told you today they are run in reverse asymmetric mode, where the support structure faces the feed, and they're run in dead end mode, okay? So why do we do this? Well, a couple of reasons. One is that you can see here, it is impractical, right, to regenerate virus filtration membranes like sterile filters. It's impractical. You can't do it because how are you going to validate that you've removed all the virus particles? So if that's the case, dead end mode is much simpler to run than tangential flow. Tangential flow is if you want to reuse the membrane. You save on pump, you save on many things. So just run it in dead end mode. That's number one. And number two is that if you run it with the support structure facing the feed stream, the support structure acts as an inline pre-filter, as we have discussed earlier, right? And since you're not going to recover this and reuse it, who cares if it fouls, right? You're not going to try and regenerate it. So that's why they are now run in this opposite way. I've said here, normal flow filtration is simpler to run, lower capital cost. Virus filtration occurs close to the end of the purification stream, as I pointed out earlier. Therefore, the feed stream is very poor, and actually fouling is due to product aggregates and variants, as I mentioned earlier. The more open pore side of the membrane faces the feed stream, acting as an inline pre-filter, allowing protein aggregates and other large foulants to be captured within the microporous structure. Very similar to biooptimal that we talked about the other day. Okay, let's move on. So virus retention is characterized in terms of log removal, or what's typically called LRV, okay? And LRV is defined as the log to base 10 of the ratio of the viral concentration in the feed to that in the filtrate. So LRV is negative log to base 10 of S, where S is the sieving coefficient for the virus. In other words, the filtrate concentration divided by the feed concentration. And it's negative because this ratio comes out negative, right, when you take the log. Okay. Total LRV depends on the nature and potential for viral contamination of starting material. So this is a calculation you would have to do depending on the drug you're making, the cell line you're making, and so forth. Adventitious virus clearance, that's parvovirus, about 10 to the 6. Retrovirus you have to calculate, okay? And there are rules, like there should be less than, say, 1 virus particle per dose equivalent of the drug. And then you can work backwards from that and say, okay, I had this huge bioreactor, how many viruses do I have to remove, okay? Biologicals produced from cell lines containing retroviruses will require high LRV. Virus filtration steps are typically designed to provide at least a minimum of 4 log removal per step. If you cannot get more than 4, if you're getting like 1 log, actually it's not worth it. Because there's another issue that comes in here. The way you validate, did anyone think of how would you validate virus clearance, by the way? So we've talked about you need all this clearance. How would you determine it? Anybody? I mean, you're not going to use absorbance you're not going to use spectrophotometer, right? That's what we typically use with proteins. Do you know how you would measure how many virus particles are around? Nobody knows? So nobody's a microbiologist. You use it by infectivity assay. You take cells that can be infected by that virus, you bring them into contact with that, and you determine how many infected cells you have. And you typically do many replicates, and you do some sort of averaging, okay? But that's how you would do it. But you could imagine that an infectivity assay is costly and very time-consuming because you have to grow these cells, you have to conduct the infection, you typically do lots of dilutions, and you get to the point where there's no more infection, and then you can determine the number of virus particles you have. So, and it's a statistical method. That is ultimately you have to do that. There are other ways you can use PCR methods, polymerase chain reaction methods. You should know that from COVID-19. So basically you're taking the viral DNA or RNA trying to amplify it. These methods are not as accurate simply because there's DNA floating around everywhere. So you might be amplifying that, okay? So you've got to be very careful. Now, if your method is specific enough, you wouldn't, but you could. The other thing is, viruses are not always perfect. You know, there are defective virus particles, there are broken virus particles, and so their DNA might be released anyway. And if you amplify that, that is not really helping you because you want to know about rejection of actual virus particles, not naked DNA as it's called. So that's why the actual infectivity assay is the best because that tells you how many of these viruses are viable. They are actually capable of infecting a cell. But anyway, it's a complicated process, okay? So the point here is that the accuracy of these assays is not great, okay? It might be half a log. So if you only got 1 log clearance, it's pointless. You know, nobody's going to accept this. This may be within experimental error, all right? There's also a detection limit. If you're below the detection limit, you can't say anything, okay? You can just say there may be virus there, but I cannot detect them, you see? So it's another problem. So that's why you typically want a minimum of 4 log, okay? Viral clearance studies are performed by spiking high-titer infectious viruses with different physical characteristics into scaled-down modules and determining the LRV. We do a lot of that in my lab, so that's something we do a lot of, but it's a tricky experiment because you could imagine these virus filters I told you are used towards the end of the purification stream where the product is very pure and you don't want any fouling of the membrane, right? Because if you get host cell proteins, host cell DNA, from the cell, because you've got to grow the virus, right? So from those cells, if they come and foul the membrane, then that is not representative of the manufacturing process, right? Because this would not be in the manufacturing process. So you will get results that are not relevant. So it's very important that your virus titer that you make is very, very pure, and that is a major challenge for these spiking studies. So it's quite a complicated area, okay, when you're doing these. Removal of both enveloped and non-enveloped viruses is required. Okay, let's move on. Common model viruses include animal parvoviruses. We work a lot with this one that is called MVM, or minute virus of mice. That's a parvovirus in mice. but there's also other ones. There's poliovirus, SD40, Synvis virus, reovirus. I told you human hepatitis B is a parvovirus in the same family. Okay, it's the same family of viruses. Non-enveloped, about 18 to 22 nanometers inside. Retroviruses are designed for larger filters, but I told you now they're removed by the same filter, so this is what used to happen. But if you went to a manufacturing facility, you may see this. Because the other complicated thing in the biotech industry is you have to get your manufacturing process validated, approved by the regulatory authorities. Once that is done, as an engineer or working there, you wouldn't want to change anything. Because even if you can simplify it by removing a retrovirus filter, now you've got to do all these validating studies again, and the regulatory authority has to come and approve it again. And that is often more costly than simply leaving it the way it is, you see. So you will find retrovirus filters in use today in older facilities, okay? And until they are shut down, it will continue, okay? So that's an interesting thing about this area. But anyway, retrovirus filters remove larger viruses greater than 50 nanometers in size, while parvovirus filters remove these smaller ones. Fouling phenomena. So fouling during virus filtration is dominated by protein aggregates, DNA, partial DNA product, or other debris. But these are product-related debris, okay? They are not cells or anything else. Significant problem in spiking studies, since in these spiking studies, virus preparations are often quite what I call dirty. In other words, they contain host cell DNA, host cell proteins, other contaminants, which can foul the membrane and lead to very different characteristics. That's why I told you spiking studies are quite difficult because you've got to have very, very pure virus titer. In fact, typically how we would do that is we would take a virus filter, we would have buffer, some sort of PBS, citrate buffer, something, you'd spike the virus into that and run it. There should be no difference in the performance of this membrane in small scale when you go with just pure buffer and just buffer with virus. If there is a difference, it's most likely due to contaminants from the virus. Because as I told you, the concentration of virus particles is low. There should not be a big difference, even though they're being rejected. Okay? That's typically how you would check this. Fouling can be significantly reduced using appropriate pre-filters, scale-up of virus filters, use the same basic strategy as sterile filtration. Okay, so a virus pre-filter is an interesting area. How much time do we have? A few minutes, okay. So if you have your virus filter here, okay, so the feed comes like this. Typically, you have an inline pre-filter. And then you have your virus filter. And in a spiking study, you would then collect your permeate here. Okay, so the pre-filter is interesting. We talked about this yesterday. We said basically you always have a sterile filter in any process, right? So the simplest type of pre-filter would be, say, a 0.1 or a 0.2 micron sterile filter. Okay, that's the simplest. Would you want to use anything tighter than 0.1 micron? The answer is no, because you'll start rejecting virus particles, right? You don't want to do that. This is to pre-filter your product, okay? And in fact, so that's the first point. The second point is product aggregates, as I told you, product variants can cause problems. And so the manufacturers of virus filters now make pre-filters that are specific for their virus filter, and they use some of the principles we talked about yesterday for depth filtration. So they might have cation groups, anion groups, hydrophobic groups, diatomaceous earth. It's like a mini depth filter, okay, to remove product-related variants from the process stream, okay? So when you start to do that, then you put a pre-filter here and you bring your virus spike in after the pre-filter. Because if you have to start to have such a complicated pre-filter, it is likely you will lose virus particles by surface interaction, not by size exclusion, surface interaction. So typically now what happens is you use a more advanced pre-filter, and things like Vipro Shield made by Milipo and so forth, and then you have here a 0.2 micron filter here, and then your virus spike comes in, okay? So that's typically what's done. And actually we do quite a lot of work on pre-filters because designing pre-filters is quite complex because many of these product-related aggregates have a significant effect on the virus filter. The major problem with the virus filter is capacity, okay? If you start to lose capacity, the whole thing ends because the virus filter has to treat the whole batch. You can't change it in the middle, okay? Scale up of virus filters typically use the blocking models that we talked about. That's what people use, okay? Whether they are appropriate or not is another matter, but that's what's used, okay? And we've discussed why there could be errors with that. Okay. Types of membranes. Let me do this. So types of membranes. So Millipore manufactures a number of skinned composite flat sheet membranes which avoid the formation of macrovoids. Initially, membranes were hydrophilized PVDF. So hydrophilized just means they're made hydrophilic with a surface coating, designed to pass proteins 70 to 180 kilodalton molecular weight. So you had Virusol 70 and Virusol 180, designed for 70 and 180 kilodalton proteins to go through. But those are some of the older membranes, okay? Composite structure features a thin asymmetric ultrafiltration layer cast on a microfiltration membrane. The pore size in the ultrafiltration layer gradually increases from the skin, progressing towards the microfiltration layer. Today, they are all inverse asymmetric structures. So one way this is done today, okay, is that here is your skin layer, and then here's your support structure, like this, right? So this has the big pores like this, and this pore size kind of is larger here, it's smaller here, okay? So it's exactly an asymmetric structure. So that's what Millipore does. Paul developed a slightly different type of membrane. Part of it was to avoid Millipore patterns. So they actually used a symmetric membrane. It's the same idea. You still have a support structure and a barrier layer, but instead of the support structure being asymmetric, the support structure is symmetric, okay? It's not exactly symmetric, but it's more symmetric. And so they have these membranes called DV50 and DV20. DV50 was for retrovirus, DV20 for parvovirus, okay? And they're normal flow filters and consists of different layers. You have multiple layers of these membranes, okay? And they were made from PVDF, okay, hydrophilized PVDF. So the DV50 is designed to remove retrovirus, more than 50 nanometers in size, while the DV20 provides significant removal of smaller parvoviruses. More recently, then Millipore also started developing their normal flow filters. So initially, it was tangential flow, but we don't do that now. It's much simpler this way. Multi-layer structures. So there's a parvovirus one and retrovirus one, but today they're all parvovirus filters. The other major company is Asahi Kasei, which makes so-called Planova 15N, 20N, 35N, and now they also make something called BioEX. But these are basically as follows. So the 35N nominally, but again, this shows how different membrane manufacturers have different ways of rating these. 35N nominally means 35 nanometer pore size, but it's actually very similar to the Paul DV50, and they claim it's 50, okay? So it depends how on the manufacturer. 15 and 20N, 15N is an older filter, 20N is the more recent one. They are both designed for parvovirus removal. The difference is the 20N is a much more permeable membrane, so you get much higher fluxes. They are all made from regenerated cellulose, okay? So Planova filters are among the first membranes specifically designed for virus filtration. That is true. They're some of the first, before Millipore and Paul, and they were actually designed for plasma fractionation, not monoclonal antibodies initially. And they're very low fouling filters, and they're all run in normal flow mode. Optimization involves maintaining maximum virus clearance, high flux, and high capacity, right? You've got to maintain your virus clearance, you want high flux, and you want high capacity, right? You want to process a lot of fluid. Existing manufacturing trains, virus filters are divided into two categories. One category used for retroviruses, and the second category for parvoviruses. But as I said today, basically, sorry, I forgot to change this. As I said today, basically, they're all using the parvovirus filter, okay? I am wondering whether I should stop there because I have a couple more things to do. Let me stop at that point. Okay, one more slide. So important to perform development studies using the same pre-filter if used, because if you don't use the same pre-filter, you could have very different performance, right? Remember, this is scaled down where you want to scale up. So you should use the same pre-filter. So I'm going to stop after this slide, and I'll finish the rest later, because I think it's just about, it's 10 o'clock, 11 o'clock, yeah. The feed stream must be representative of the industrial scale processing, including both storage time, storage temperature, as well as process holes. And let me explain that. So this is very important. remember, this is one of the few cases, and for engineers this is important, where we're scaling down. So it's very important if you're scaling down that this is representative of your manufacturing scale, okay? You can't do something different. So typically, how would you run a virus filter? So if you think of it, you have the virus filter, you've got a product coming through, it's a dead-end filter, you run it, okay? Maybe you run it for 4 hours, I don't know, 5 hours, then you stop. Now, your product is very valuable at this stage. It's towards the end of the purification strain, okay? Every milligram could be even worth $1 million. It's a lot of money, so you want maximum recovery. Now, as you know, the tubing, all of this will have your product there, right? Because you're pumping from a container through this filter. You want to recover all that, otherwise it's just sitting in the tubing. How do you do that? You do something called a buffer flush. You flush with the same buffer, all right? And you flush it through and you collect it. You dilute your product a bit, but remember in our diagram it didn't matter because at the very end we had a concentration step, diafiltration, so that's fine. The problem is this, that it has been shown that if you have a normal flow filter like this, okay, and you look at the structure, so the virus particles are trapped here in pores. You have to, when you switch to a buffer, you have to stop. You release the pressure, okay. This is running at, say, typically maybe 1 atmosphere, the maximum maybe 3 atmospheres. It's not very high pressure, okay, maybe 3 atmospheres. You have to release the pressure. As soon as you release the pressure, the viruses may be trapped in pores. They can actually back diffuse, right? There's a possibility for Brownian back diffusion if we release the pressure, and then they can find other pores that may be more open and they might get through, okay? And it has been shown that when you have this pressure release, in other words, a hold while you're switching over, that actually you suddenly find virus coming through. Not many, maybe a few, but you're trying to get huge removals here. So this is a big problem, okay? So this pressure hold is a big problem. So that's why I'm talking about here, including both storage time, storage temperature, as well as pressure holds, all have a significant effect on capacity. So you must make sure when you do your scale down study that you have the same pressure holds. So if you're going to pause for 30 minutes, you should do the same thing again. It's very important you do exactly the same thing, okay? It's important to extend the capacity study beyond the throughput in the manufacturing process, maybe 1.5 to 2 times to deal with feed stream and membrane lot-to-lot variability. But that's a typical thing engineers do, right? We like to have factors of safety, okay? So I'm going to stop at that and then let Professor Chidham continue with his talk. You're next, right, I think? Yeah, he's next. You're next, right? So, there's a tea break before that. Okay, so let me stop. Let's have our tea break. He will continue, and then in the afternoon, I have a few more slides on this, which we'll finish off with, and then we'll move on to membrane adsorbers. Okay? All right. Thank you.